Hello, my lovely anatomists and physiologists. Welcome back to another chapter 12. We're getting close to the end of our conversation here. What we're looking at in this video is how do you actually get that neurotransmitter to be released from that synaptic knob? What are the actual steps that are involved there? So before we kind of get into that part of the conversation, let's go back and talk about there are two different kinds of synapses. There's two different types of synapses. You can have either an electrical synapse or you can have a chemical synapse. In the case of electrical synapse, this is where you have a direct connection between membranes of two different cells. And so ions can move directly from one cell to the next. So essentially, if cell A is depolarized, then cell B will also be depolarized. So in the case of an electrical connection, the membranes are connected, touching each other. There's no synaptic cleft. And, it's basically kind of a synchrony of one of the cells connected is depolarized and the other cell will be too. The chemical style of synapse involves a neurotransmitter and a synaptic cleft. And this is what we've been studying so far. So we've been dealing with neurotransmitters being released at a synapse, at specifically a chemical synapse. Now, in the case of a chemical synapse, you're going to have, let's add some detail here. You're going to have a presynaptic, we can call this element. This is typically a cell. And for us, I think we should think about that as the synaptic knob. So if you look at the diagram that I've brought into the presentation from our OpenStax Anatomy and Physiology textbook, and we've been drawing this synaptic knob and thinking about it since chapter 10. This is the very end of the axon terminal. You have that enlarged part of the axon terminal, and it's full of that neurotransmitter. Obviously, we need to have a neurotransmitter. We've been focusing in on the neurotransmitter acetylcholine, but there are, of course, lots of others in the body. So we'll be adding to that body of knowledge as we move through the course. I already mentioned up above that we're going to have a synaptic cleft. That's part of a chemical synapse. We're going to need to have some receptor proteins. So we see those, we saw those either at the motor end plate when we were looking in chapter 10. We see it in our diagram over here as part of that postsynaptic neuron. So maybe located, as part of the dendrite or there at the axon hillock. These are our chemically gated sodium channels. Remember, we can also use the term ligand gated. So that is there in our diagram. So I'll highlight that for you. We can actually also use the description of a nicotinic receptor, and it's talking about the same actual type of receptor protein. We need to have some kind of post-synaptic element. So in our diagram, it's a postsynaptic neuron. In chapter 10, that was the motor end plate on the skeletal muscle cell. And then you have to have some way of actually removing the neurotransmitter from the cleft, which we didn't actually talk a lot about in chapter 10, although we could have. So you need to have a neurotransmitter elimination or uptake. And in the case of acetylcholine, we see acetylcholine ester ACE, which is abbreviated big H, big C, little h, big E. If you go back and you look at your materials for chapter 10, maybe you noticed there were these little crescent moons that were like hanging out inside that synaptic cleft. That little crescent moon was representing the acetylcholinesterase. So this is the enzyme that will break down acetylcholine in the synaptic cleft, and then those components get taken back up by the synaptic knob and get used in making of new acetylcholine. So you had to have a way of like stopping the signal. So when we actually look at the neurotransmitter release, we're seeing the picture again from our OpenStax anatomy and physiology textbook. So take a look at that diagram. We see we're looking at a presynaptic neuron synapsing onto dendrites of a postsynaptic neuron. And we see that the presynaptic neuron has that synaptic knob. It's got those vesicles of acetylcholine. We can see the synaptic cleft where the acetylcholine will be released into. We can see located on the postsynaptic neuron, those ligand-gated channels are what we can call chemically gated sodium channels. There's just a couple of details that we haven't really put into our conversation that we need to add to our conversation now. I think I have enough room just to kind of build that in down here. So what we're going to see is some of the story we already know. Let's start with the action potential reaches the synaptic knob. And I'm going to put in parentheses that that is our presynaptic. I'm going to say neuron. We could say element, which is what we have in the description up above. That's coming in our diagram there from that presynaptic neuron. When the action potential reaches the synaptic knob, that actually triggers the opening of voltage-gated calcium ion channels. Voltage-gated calcium ion channels. And when those channels open, we see calcium ion floods, synaptic knob. And this triggers acetylcholine. I'm going to say triggers exocytosis of acetylcholine. So exocytosis, remember, is that type of vesicular transport we discussed in chapter 3, where we see the vesicles fuse with the membrane, and then the neurotransmitter in this case is released into that synaptic cleft. So the acetylcholine is then going to diffuse. We should be able to kind of follow the rest of the story. Acetylcholine will diffuse across that synaptic cleft. And it's going to bind to the chemically gated sodium channels or ligand gated channels. These are part of the postsynaptic, we could say element or neuron here. And I'm going to put at the dendrite so we kind of know where we are in that cell. Okay. So when we talk about that postsynaptic neuron getting stimulated, and we talked about, we had team acetylcholine being released and like jumping across to land in the chair, push the button. How did that neurotransmitter get released, right? And we picked that up in chapter 10. We said in chapter 10, when the action potential reaches the synaptic knob, we got exocytosis of acetylcholine. So we knew this already, but what we didn't know maybe was the role of calcium ion. So make sure that you are paying attention to the role of the calcium ion in this exocytosis. And that links us back to that conversation we had in chapter 6 about calcium regulation. The last bit of information to talk about here on this video are different neurotransmitter systems. So this is introducing the idea that we have different neurotransmitters that are involved. We have two major systems that we'll be talking about in our coursework. So we're going to be talking about cholinergic systems. And by definition, cholinergic systems involve the neurotransmitter acetylcholine. So that's what we've been talking about since chapter 10. Now we'll see that cholinergic systems have two different types of receptors. So we have the nicotinic receptor. Y'all, that is the same as our chemically gated sodium ion channel. It's the same as our ligand gated channel. These are gonna always depolarize. Because these are ion channels. So when acetylcholine binds to this receptor, they open and sodium floods the cell, and that's depolarization. Depolarization is always a stimulation. The other type of receptor is called a muscarinic receptor. Muscarinic receptors can either depolarize or hyperpolarize. This is the beauty of these receptors. The cell makes the receptors as part of the cell membrane. So that means the cell is determining through its receptors what kind of neurotransmitters it can listen to and how it responds. So any cells that have nicotinic receptors are gonna listen to acetylcholine and be stimulated by acetylcholine. But acetylcholine can then inhibit a cell that has a muscarinic receptor. So the significance here is it's not in the nature of the neurotransmitter, whether it's excitatory or inhibitory, it's in the nature of the receptor. So the receptor is actually determining how the cell responds, and the cell is making its receptors. So ultimately, the cell, through the making of its membrane and the making of its receptors, is determining like who it listens to and how it responds. The other important system that we'll talk about this semester is adrenergic. Think of adrenaline. Adrenergic systems, by definition, involve epinephrine, which is adrenaline, and its buddy norepinephrine. You'll see in the future that we'll use a big E to abbreviate epinephrine and then a capital NE to abbreviate norepinephrine. Just like cholinergic systems have two different types of receptors, adrenergic systems have two different types of receptors. So we're gonna have what's called alpha and we're gonna have what's called beta. And we're gonna get into these different receptors more in the future. So that's pretty much how we want to list that here. When we talk about the adrenergic systems, this is part of the sympathetic division. Sympathetic division was our fight, flight, or freeze. These are those responses that are important in either like increasing the metabolism of our body or just like mobilizing and protecting us from danger. So equally relevant, equal ways that our bodies respond to stress, fighting, fleeing, and freezing. So our bodies do combinations of these. That's the sympathetic division. The cholinergic systems, we'll see this in the parasympathetic division. We see it in the somatic division. And we see acetylcholine also in the sympathetic. So talking about acetylcholine as a neurotransmitter is a great starting point for us because we see it really prevalent in the body. There is one other piece of information I want to pass on here. Okay, the last piece of information has to do with those receptors. Those are membrane receptors. Remember, they determine how a cell responds and whether or not it responds. I'm just going to say chemical messenger. I mean, in this chapter, in this context, the chemical messengers we're talking about are neurotransmitters, but this information to some extent applies when we talk about hormones. meaning the membrane receptors determining whether the cell is inhibited or depolarized, stimulated by the messenger. And we have two different styles of receptors. So we have what's called the ionotropic receptors. So these are ligand gated ion channels. These are going to depolarize. This is like either nicotinic receptor or what we have always been calling the voltage gated, the chemically gated sodium channel. The other kind of category is the metabo, as in metabolic, metabotropic receptors. Let me stop just a minute and over here say, tropic is like turn on. So ionotropic is turn on by ions. Metabotropic is like turn on metabolism, but it can also turn off metabolism. These involve a transmembrane. Well, let me do this. You have a first messenger. In this case, that's our neurotransmitter because that's what we're talking about. It could be a hormone. It binds to a transmembrane, trans is across, receptor. This activates what we call a G protein, which in turn activates a second messenger. And the second messenger is usually either a cyclic AMP, which we abbreviate lowercase CAMP. This is adenosine monophosphate, but it's a circular instead of linear. Or very often our second messengers will be calcium. This is how I like to view my G protein. This is how I like to view my G protein. My G protein is this bridge between the first and the second messenger, but it's not either one of those things. So it's sort of like the kids sitting in the front room playing Xbox and somebody knocks on the front door and they're closest to the front door, but instead of maybe stopping the game and opening the door, they just yell like, "Hey parent, somebody's at the door." And you have to stop what you're doing and walk to the front room and see who's at the door and all that, right? So in that case, like the person knocking on the door would be like the first messenger. That's the neurotransmitter that's binding to that receptor. The G protein is like that teenager who's just letting you know somebody's there. So they're helping, but not really. And then the second messenger would be like, okay, the parent who, is actually doing the response, right? So it's actually changing the cell, and the cell can then be stimulated or inhibited, again, dependent on the receptor and the G protein and the second messenger. But we get some kind of change happening in the cell. All right, y'all. I added this last little piece so that we could finish with this last video. So take care of yourselves and each other.